So in the first question, in that paragraph at the top, I tell you about pure breeding bunnies. That's what Mendel did. He had pure breeding plants. And his definition of dominant and recessive was about what showed up in the F1 generation. So can somebody tell me which color is dominant? Yeah. Black. Black is dominant because the F1 generation, it says, have black fur. Now, since I started with pure breeding bunnies, I can use this definition. If I hadn't started with pure breeding bunnies, I can't guarantee that black is dominant, but based on that context, this sounds exactly like what Mendel said, and so since the F1 generation all had black fur, I can conclude that black was dominant. Now, when I'm making my predictions for the possible phenotypes of the offspring, the first step is always to figure out what's the genotype and phenotype of the parents, right? That's step one in doing a cross. So it tells me right in the context that there is a black-furred bunny, and it says that that black-furred bunny is heterozygous. So I might write big B, little b as the genotype for my heterozygous black-furred bunny. It's not super important that you chose the letter B, but rather it's important that you had a capital and a lowercase letter. Now, all I told you was that I crossed it with a white-furred bunny. I didn't say what the white-furred bunny's genotype is, but you should be able to tell what the white-furred bunny's genotype is. Does anyone know what the genotype of the white-furred bunny is? Right, it has to be homozygous because it's the recessive trait. So if you were using the letter B, you probably put two lowercase b's. So that would be the white-furred bunny's genotype. Anytime an organism has a recessive trait in this dominance scenario, they have to be homozygous for the trait. If they had even one copy of the dominant allele, that would be the one that's expressed. So you're making a Punnett square with big B, little b, crossed with little b, little b. You might have realized, hey, the white-furred bunny can only make one type of gamete. I don't even need a 4-square Punnett square. I could get away with a 2-squared Punnett square. Now I'm just going to sketch this out on the whiteboard because when I'm on a Google slide, the pen does not line up nicely. But my Punnett square would have looked something like this. And then I could have said, oh, I don't need this second one here because it's the same as the first one. I would have ended up with one box giving me a heterozygous genotype, so black fur, and one box giving me homozygous recessive, so white fur. So my response to this question would be that 50% of the offspring should be black and 50% of the offspring should be white. If you have that, then you are correct. Let's say we'll give 3 marks total for this question. If, let's say, you messed up on which one was dominant, but then you did your cross correctly, I'd probably give you 2 out of 3. If you had the correct dominance, but maybe you messed up somewhere on your Punnett score, I'd still probably give you 2 out of 3. If you're not sure, just leave it. I'm going to read everybody's responses anyways. Does anyone want to ask about that question? Okay, so number 2 was probably fairly straightforward if you did the practice questions, because one of the textbook questions was basically this exact thing. Mendel has this plant. He knows that it has the dominant phenotype, but he doesn't know if it's homozygous or heterozygous. So what should he do? And I can see quite a few of you have the correct response here. Does anyone want to read what they wrote for this? Okay, I'm going to ask maybe Nicola, can you read yours? Because I read it and it was correct.
Okay, so if you cross the tall pecan with the dwarf plant and any of the F1 generation genotypes are like the dwarf ones, then it means that it's heterozygous.
Very nice. So she said, sorry, I'm just going to repeat. She said you take this plant and cross it with a dwarf plant because that's the recessive trait. And then if any of the F1 generation plants show that dwarf trait, it means that my original plant was heterozygous. My example yesterday in the notes was with the Walter mice, but it's that same exact idea. So if you described a test cross and what you should do in a test cross, you are correct. What I want to be want you to be careful of is if I were to ask you this type of thing, say on a unit test or something as a written question, I'm not looking for a number as the answer. I'm not looking for something like 50% of the offspring will have this trait, because I didn't actually give you any data. I'm just looking for the steps. And so what Nicola read out and then I repeated, those are the steps. That's what you should do. But we're not looking for a number or a ratio as the answer. Now we'll say this one is out of 2. So if you knew that it was a test cross but maybe didn't quite explain exactly what that was, I would probably give you 1 out of 2. If you explained that you would have to cross it with a homozygous recessive organism and then look at the offspring to see what happened, you've probably got both ideas, and I would probably give you 2 out of 2. And if you're not sure, so we're going to move on and talk about some different scenarios. So we've got that first type of cross. We know what a test cross is. We understand how Mendel figured out dominance in his pea plants. So now we move on to another of Mendel's laws. I'm referencing a page in the digital textbook, so of course you could look that up if you want to see a picture of it. But when I tell you this, you should be like, hey, didn't we already learn this law? And I'll be like, yeah, because we talked about it during meiosis. It's the law of independent assortment. So you should already know what independent assortment is, because we already talked about it, but in the context of genetics, Mendel had something to say as well. During the formation of gametes, which he mentioned in his first law, there's segregation during the formation of gametes, the 2 alleles for one gene separate or assort independently of the alleles for other genes if, and this is super important, they are on separate chromosomes. We will see the scenario of if they're on the same chromosome in the next lesson, but for now, if these genes are not one, but on separate chromosomes, then the genes will assort independently because chromosomes assort independently during gamete formation in meiosis. So in Mendel's world, he did something called a dihybrid cross. So first it was monohybrid, and now it's dihybrid. The prefix di means 2. So now instead of crossing organisms on one trait, we're going to cross them on 2 traits. So the description is that it's the crossing of 2 pure breeding plants, and Mendel was always using these pure breeding plants when he was trying to figure out which trait was dominant and which trait was recessive and how things were passed on. The definition of pure breeding doesn't change. It means they're homozygous for the trait that we're studying, but this time it's for 2 traits. And he was trying to determine if one allele influences another. And so I'll just repeat again, in what we're going to look at today, the alleles don't influence each other. Independent assortment happens. But as a bit of suspense for future lessons, they can. It's just that in this first scenario that Mendel was looking at, they didn't. Dihybrid crosses can involve heterozygous organisms as well, but Mendel always started with pure breeding plants at the beginning, and I just needed to make sure you were clear that just because that's what he started with, it doesn't mean that's what all crosses have to start with. Now, before we go through some examples of how it might look in a dihybrid cross, does anyone want to ask anything about independent assortment or what Mendel was doing in his dihybrid crosses? Okay, so when we look at the examples, we're going to be referencing the same traits as we were before. Things like purple or white flowers, tall or short plants, green or yellow seeds. So the same traits we were looking at before. So if you printed out the notes, there's that little table at the beginning that showed all of the traits that Mendel studied. We're looking at the same traits, but we're just going to look at two of them at a time now. Whoops. So you'll notice that my blank Punnett square just got a whole heck of a lot bigger. It doesn't mean that every time you do a dihybrid cross, you need a 16 square Punnett square, but this would be like the maximum size that we could ask you to use. Of course, in real life, I could ask you to do a trihybrid cross, but I won't. So for the purposes of our class, this will be the maximum size of Punnett square that you would ever need to use. So we'll start with a little example from Mendel. Mendel did a test where he crossed tall green plants that were pure breeding for both of those traits with short yellow plants, again pure breeding for both of those recessive traits. So the capital letters were the tall green plant, the lowercase letters were the short yellow plant. Those are the same traits that we looked at in examples yesterday. Now, without making a Punnett square necessarily, I should know that the F1 generation in this scenario of complete dominance should all be tall and green. I could make a Punnett square, but it would only have one box. My tall green plant could only make gametes that have the tall allele and the green allele. So on one side of my Punnett square, I would have capital T, capital G. I've got two letters this time because I'm looking at two genes, not just one. My short yellow plant, because it's homozygous, could only make a gamete that has the recessive allele for height and the recessive allele for color. So my short yellow plant could only make a gamete that has the short allele and the yellow allele. Again, two letters because I'm looking at two genes. When I fill in the box for something that's based on two traits, I'm going to have more letters. All of the F1 generation should have one capital T, got it from this allele, or from this parent, one lowercase t, so one dominant, one recessive allele for height, one dominant allele for color, and one recessive allele for color. Now, just like in the examples yesterday, I always write the capital letter first, because that's how words look, and that's how you'll see it depicted on test questions. The capital letter always comes first. And I always group like letters together. So I put the two T's next to each other. That way, when I look at my Punnett square, I'll say, oh, there's a capital and a little t. That means dominant is the trait that's going to show up. If I didn't put them in order like that, it might be harder for me to correctly interpret which phenotype I'm going to get. So I always group the letters together so that I have both alleles for one gene together. Now, we are going to make an F2 generation from this F1 generation. When Mendel made an F2 generation, he always crossed two plants from the F1 generation. We just decided that the F1 generation were all going to have the same genotype. There was really only one possibility for them. So the cross that we are going to do is this. Okay, so that's the two parents that we have. So we have to consider now that there are multiple different possibilities for the gametes that these parents could make. We just said that independent assortment is happening. We're going to assume in this example here, even though it doesn't say, that they're on different chromosomes. And in fact, you can assume that unless the question tells you otherwise, because that is the standard scenario. Lately on diploma questions, they've been adding a little disclaimer that says the chromosome or the genes are on separate chromosomes so that you're sure and confident. Now, when I consider the genotype where the parent is heterozygous for both traits, here are all the possible gametes they can make. Each gamete has to have one of each letter. So each gamete has to have an allele for each gene. So one possibility could be that the gamete gets the dominant allele for height and the dominant allele for color. Or dominant allele for height, but recessive allele for color. So capital T, little g. I could also make a gamete that has recessive allele for height, dominant allele for color. So little t, big G. And then I could make one more, I'll just do a little circle here, that has the recessive allele for both traits. Little t, little g. So notice, each gamete had one allele for each gene. Not two alleles for the same gene, one allele for each gene. Always one allele for each gene. The only thing that's different here is that I have two genes that I'm considering instead of one. So I have multiple combinations that are possible. If a parent is heterozygous for both traits, 4 is the number of gametes you'll have. Now this would be the most. It's possible that a parent was homozygous for one of the traits, in which case you'd have less gamete possibilities, but I'm showing you the maximum here. Since the other parent had the same genotype, I'm going to fill in the same 4 possibilities on the other side of my Punnett square. And now when I fill in my Punnett square, I have to recognize that I have 2 genes, and so I'm going to do what I did when I made the F1. I'm going to group the letters together, keep T's together, keep G's together. I'm going to put the capital letter first if it shows up. So I'd like for you to take a minute here, because it will take probably more than a minute. Now if you want to, you can look at mine that I filled in up here. Very often, especially when we have this many possibilities, we'll focus on the phenotypic ratio rather than the genotypic ratio. The reason being there are so many possible genotypes, but a few less possible phenotypes. Now I stated over here that a pretty standard ratio in a lot of Mendel's experiments with these pea plants was that in the F2 generation, he would get a ratio of 9 to 3 to 3 to 1. And I want to show you what that represents. The first number is the 9. The 9 represents organisms who have both dominant traits. So in this scenario, it would represent tall, green plants. So if I count in my Punnett square, and this is somewhere where I find color coding super helps me. I'm going to see if I can hopefully change my pen color here. But I'll often do this, especially when I have a bunch of things that I'm trying to sort through. I'm going to circle all the ones on my Punnett square that give me tall and green. This one will. So will this one, and this one, and this one. Every single thing in that first column has at least one capital T, so it's tall, and at least one capital G, so it's green. If I go across this top row, I'll find some more. And again, I'm looking for at least one capital T and a capital G together. In the second row, this one will give me a tall green plant. The other two won't, though. This one will give me a tall green plant. Now, if you count, 4 in the first row, 2 in the second, 2 in the third, 1 more in the fourth row, that makes 9. 9 of the 16 possibilities in this Punnett square gave me a tall green plant. That was both dominant traits. So I'll just make this. And then we'll switch colors. Now the next possibility, the next number in the ratio, are plants that have one dominant and one recessive trait. So I should find 3 plants that are tall but that are yellow. That means they have a capital T in their genotype, so they have the dominant allele for height, but they don't have a capital G, so they don't have the dominant allele for color, meaning they have to have the recessive phenotype. So here, and here, and here, 1, 2, 3, are 3 possibilities in my Punnett square that gave me a tall plant, dominant allele for height, but no dominant allele for color, so they ended up being yellow. Let's pick maybe this bluish green. Now the next 3 in the ratio represents sort of the opposite. Plants that have one dominant and one recessive trait, but the other way around. So my first 3 had dominant allele for height, recessive allele for color. My next 3 is going to have recessive allele for height, dominant allele for color. So I'll see that 1, 2, 3 plants would give me that scenario. I will have 3 dwarf or short plants that are green. And then the last number in my ratio, so I'll use this one, is this one. This is the one that has both recessive traits. And in this example, dwarf and yellow. You will see this ratio come up again and again, just like you'll see the 3-to-1 ratio that we talked about yesterday, and another one that we'll talk about tomorrow. If I have 2 heterozygous organisms based on 2 genes and I cross them, I should get a 9-to-3 to 3-to-1 ratio. And knowing that could be the way that you respond to a question, right? Because this is a predicted known ratio. And sometimes if you can see that that's the ratio, it saves you a lot of time and a lot of work. Now, would anyone like to ask anything here? Yes, Rose. The turquoise one here was dwarf green plants. So they had the recessive allele for height, but the dominant allele for color. And so these 3 boxes here all had little t's, so they were short, the dwarf plant, but at least 1 capital G, so they had the dominant allele for color. Okay, so we're going to do another example. And in the next example, I'm asking you to cross 2 plants. I didn't tell you what their phenotype is. I told you straight out what their genotype is. So maybe a bit simpler than your typical test question, because you don't have to figure that out. I put a 16 square Punnett square there, but you may not need to use the whole thing, just as a little preview. And so I'd like you to figure out the phenotypic ratio in the offspring if those are the 2 plants that I'm crossing. It'll be different than the last example because my 2 plants are not both heterozygous for both traits. So folks, if you're feeling stuck, I wrote what the gametes these 2 parents could make around the outside of the Punnett square so that you could check and see if that's what you had. And I also indicated along the left side that for one of the parents, I would be repeating myself if I made 4 different possibilities for the gametes, so you can omit those 2. But I'll wait another couple of minutes for people to give it a try with this, and then we'll go through it. All right, I think we'll talk about this so that we still have time for you to look at those practice problems for lesson 2 that are in your handout. If I take a look at my Punnett square, my first step is always figure out what's the genotype of my parents, but that's already done for you here. If I wanted to know their phenotype, I would say that this first parent was tall and green, and the second parent was dwarf and green. But I really needed the genotype because I used the genotype to make the gametes. So if I consider this first parent who has a heterozygous genotype, we're going to follow the same pattern we did for the last example. There are 4 different gamete possibilities. Every gamete has to have 1 and only 1 allele for each gene. So you would never put 2 T's together or 2 G's together. You would always have 1 of each letter. So it's the same 4 possibilities from the last example. The other parent, though, doesn't have 4 distinct gamete possibilities. The other parent is homozygous recessive for the height allele. It only has little t's, the lowercase t. And so if I think about what gametes it can make this little t with a big G, or this little t with a little g. Or it could make this little t with a big G, this one with a little g. It's the same thing. I would be filling in the exact same thing in rows 3 and 4 of my Punnett square as I did in rows 1 and 2. So I'm going to recognize that there are only 2 distinct gamete possibilities, and I'm going to save myself some time. What you'll see is you'll end up with the same overall ratio, whether you do all 16 squares or just the 8 that I filled in. Now I've written down here the same ratio that we looked at in the last question. We'll start with the organisms that have both dominant traits, then ones that are tall and yellow, then ones that are dwarf and green, so one dominant, one recessive trait. Then we'll go with how many of them have both recessive traits. Now here's something that's kind of important. Most of the time when we're looking at ratios, we are looking at the most simplified version of the ratio. So when I count how many organisms have each trait and I make my ratio, recognize it would have been twice as much if I had filled in all 16 squares. So if I look for tall and green, tall green, tall green, well that was a fun thing, tall green here. Okay, so I have 1, 2, this should have been a full circle, but I have 3 of them that are giving me tall and green, both dominant traits. Then if I look at my next number, I want ones that are tall but yellow. So here's tall and yellow, because I have a dominant allele for height but not for color. Then my next one is looking at dwarf and green. So I'll pick this kind of turquoise color. This one will give me dwarf and green. So will this one. So will this one. They all have 2 copies of the recessive allele for height, but at least 1 copy of the dominant allele for color. And then my last box will be the one that has both recessive traits. So the dwarf yellow. So notice how my ratio changed. 3 to 1 to 3 to 1. And we might inverse that 1 and 3, right? There's not a set order that you have to put things in. But it wasn't 9 to 3 to 3 to 1. We didn't have 16 distinct possibilities. We only had 8 in this scenario with the different gametes we could make. So 9 to 3 to 3 to 1 is a ratio that you can expect when you have 2 heterozygous parents for 2 traits. But as soon as one of the parents is no longer heterozygous, is homozygous for a trait, then our offspring ratio is different. If you had filled in all 16 squares of the Punnett square, your ratio would be 6 to 2 to 6 to 2. And now, normally I go and I rip the diploma exam apart before I let them show it to you, but they have not had a lot of let Mrs. Baxter look at the diploma exam and judge it before you guys write it. But in recent years, they have been what I would say is more flexible about what type of ratios they accept. But if it were me, I would always, always, always give this most simplified ratio because that is guaranteed to be a correct answer. Now, on your practice